So I'm going to be talking to you about how genomics is applied to fungal research with a main focus on fungal pathogens. So here we go. So the WHO defines genomics as the study of the total or part of the genetic or epigenetic sequence information of organisms and attempts to understand the structure and function of these sequences and of downstream biological products. So in the past quarter century, the cost of sequencing has fallen dramatically. So if we look at this graph here, the cost of sequencing a full human genome has fallen really rapidly. And as such, fungi has also followed. So fungal genomes are considerably smaller than human genomes. So depending on the size and which technology you're using, whether it's short read or long read, you can probably sequence a fungal genome for about £100 or less, which has made it a lot more accessible for us in terms of research. But just how useful is genomics in mycology? Well, in terms of studying fungal pathogens, the majority of research is focused on using genomics to find out why and how fungi are pathogenic. And we can do this by comparing genomes to identify mutations caused by single nucleotide polymorphisms, SNPs, that contribute to drug resistance. We can compare genomes to identify when drug-resistant strains are spreading, for instance, within hospitals, but also on a larger scale across the globe. We can study the evolution of genes involved in host adaptation. And some fungi are endemic to specific regions of the world and cause quite severe infections. So thinking blastomyces, histoplasma, coccidioides and paracoccidioides, sporotrichs. Genomics can be used to study how these species differ from non-pathogenic varieties and how they've evolved to infect humans. Genomics can also be used in diagnostics and then also surveillance. So I'm going to talk a little bit about how we've used genomics in terms of surveillance of outbreaks using the kakapo parrot as an example a bit later on. And of course, Matt's already touched on this a little bit. Genomics has also been used quite widely in the use of model organisms. So Saccharomyces cerevisiae is obviously a model organism for eukaryotes, but Saccharomyces cerevisiae is an important organism in its own right because it's used in fermentation. And I think a lot of us here probably do like the products as a result of that industrial usage. Genomic sequencing and analysis has shown that there's been two separate domestication events for cerevisiae, so one for sake and one for wine. This was back in 2005, but then there was quite a beautiful seminal paper that showed that there was this out of China hypothesis. They analysed over 1,000 genomes for cerevisiae, and they saw that there was this single out of China hypothesis followed by independent domestication events. The Chinese isolates were highly diverged from the rest of phylogeny, which suggested this geographical origin, and this was supported a few years ago in another paper. But there are some really nice exceptions. So there was wild Mediterranean oak tree strains, which grouped with domesticated strains, and then also sake strains, which grouped with clades of wild isolates. So there are just really nice examples of this out of China hypothesis and how genomics can be used for non-pathogenic fungi. So we're really interested in how we can apply genomics for resistance detection, surveillance, and tracking. So fungal samples can be acquired from the clinic or the environment, including engaging with the public as citizen scientists. So for instance, here we can bring in samples from all of these different niches, perform standard microbiology and whole genome sequencing, and then perform SNP calling and phylogenetics, surveillance and transmission tracing. As humans, we are continuously exposed to aerosolized fungi, and we've also in the past set up these air filters on the rooftops of buildings in urban locations and then also in rural locations. And we've picked up a lot of Aspergillus species, predominantly Aspergillus fumigatus. Aspergillus really likes to grow up to 60 degrees C in soil and compost, and it really likes to sporulate as well, so it goes up into the air. And there's certain patient cohorts that are at risk of developing a disease called aspergillosis, which is caused by Aspergillus fumigatus. And this can be an invasive infection. So the most common cohort to suffer from this in the UK, at least, is cystic fibrosis patients. We have quite a large cohort in the UK. There are other cohorts at risk. And then more recently, we've seen severe influenza and, of course, COVID-associated aspergillosis as well. Aspergillus phenotypically is extremely diverse. So you can see Aspergillus fumigatus here in the center top. But if we look at other Aspergillus species as well, phenotypically, they are all quite diverse and quite beautiful as well. And genotypically as well, the genomics is very diverse. So Aspergillus is as diverse as our own phylum, the vertebrates. So if we consider Aspergillus fumigatus and Aspergillus fisheri, they're as divergent as humans and mice are. The genomes are quite stable. They're not what we would define as plastic, so they don't tend to display a lot of aneuploidy, which we do see in, say, Candida and Cryptococcus species. But there are a lot of differences, as we can see. And the reasons for this are pangenome, variation, but then also horizontal gene transfer. So just as a little sort of primer on the pangenome is the entire set of genes from all strains or isolates within a clade or a species. So if we take this little cartoon here, we've got 3 strains just as a representative. The yellow and the blue genes are common to all 3, so they would be defined as the core genome. But the purple, the red, and the green aren't common to all 3. So they are the accessory genome, with the green gene only found in the middle strain, so that's classed as a unique gene. If we look at Aspergillus fumigatus pangenome, the Aspergillus fumigatus, as Matt mentioned, it's a critical priority fungal pathogen with high rates of resistance. When we've studied it previously, we've used a single reference genome. And this has been annotated to have just under 10,000 genes, over 29 million base pairs. But when you look at the pangenome, there's a lot more diversity. So there was 3 papers that came out around about the same time from 3 different groups, and they all sort of found roughly the same thing, that there was actually more genes that we were missing. So there was probably about 13,000 genes in the Aspergillus fumigatus pangenome. The core genome was enriched for housekeeping functions, which is perhaps expected, but the accessory genome was super interesting. It contained enrichment for secondary metabolism genes and antifungal drug resistance. So you saw this slide on Matt's talk. Aspergillus fumigatus drug resistance is centered around a gene called CYP51A. And the most common polymorphism to cause azole resistance in Aspergillus fumigatus is a Tanner repeat of 34 base pairs in the promoter, coupled with a leucine to histidine amino acid change at codon 98. There's also another polymorphism with a Tanner repeat of 46 base pairs and 2 amino acid changes as well, but TR34 is the most common. So we've looked at sequencing a really wide spatial and temporal range of Aspergillus fumigatus isolates to try and figure out what the origins of this resistance mechanism were. So if we look here at the map, we've got quite a wide spatial range. And then at the bottom with the timeline, we've got quite a large temporal range as well. So the earliest isolates are from 1919. So we've got a temporal range of over 100 years. So when we look at the phylogeny of Aspergillus fumigatus, we can see that there's these 2 clades, as Matt mentioned. Matt said you don't really need to do the stats, and okay, yeah, sure, it's pretty clear here, but I did do the stats, and I can confirm that statistically speaking, it is well supported. The blue isolates, so the blue circles, they're TR34L98H isolates, so they are azole resistant. As you can see, the majority of them are in this bottom portion of the phylogeny. They are found globally, but they have increased in prevalence over time. These are the clade A isolates. You're just as likely to find a resistant isolate as you are a drug susceptible isolate. And then clade B is the mainly drug susceptible clade. The eagle-eyed among you will spot that there are some blue isolates in the clade B, and we think that that is through crossing and recombination has shown it on that background, but it doesn't seem to spread. So when and where did resistance originate? So we looked into the spatial temporal origins of both TR34 and TR46. TR34, through Bayesian analysis, seems to have originated in the late 1970s. And this has got really good confidence values. TR46 seems to have originated a little bit early, which was a little bit surprising because it's not as commonly found, but the confidence around this estimation is a lot broader. So actually there is an overlap here. So it could have originated later. Spatially, and I do often joke that this is why I've had to leave the Netherlands because I've had to go into witness protection. We think it could either be East England, so perhaps East Anglia, or also the Netherlands as well. And what is common to both of these areas is that it's a very agricultural niche. There's a lot of farms in the east of England, there's a lot of fruit farms. In the Netherlands, of course, there's a lot of horticulture. But what we do know is that from the environment, patients then acquire these infections via the inhalation of air. So there was a really nice study led by Jen Shelton as part of her PhD, where she embarked on this citizen science sampling program, and she combined her air samples with clinical and environmental isolates. And if you look at the genetic diversity of the air samples, you can see that generally speaking, they're all much of a muchness. They're all drawn from the same population. And then when you look at these phylogenetically, the air isolates are dispersed throughout the phylogeny with the clinical and environmental isolates. But what's really super interesting is here, if you look at the phylogeny, the air isolates are the green blocks highlighted with the red asterisks. There are some blocks that are pretty much just entirely air isolates. So we think that there's some sort of genetic diversity that we previously missed and can only be picked up by sampling the air. So it might be that there's other niches that also have a certain amount of genetic diversity that we are missing. So surveillance of fungal outbreaks is one of those things that we are quite passionate about trying to move forward. And there are existing antifungal surveillance strategies, so the Sentry program, for instance, but future surveillance priorities should really try and employ a genomic angle. And the WHO have requested genomic surveillance strategies in the aftermath of COVID. And of course, this does align quite nicely with the fungal priority pathogen list, as Matt introduced. So in light of that, we have worked with David Arnenson and his team at the Centre for Big Data in Oxford to develop Pathogen Watch, which is a global platform for genomic surveillance. And there is a Candida auris scheme on there, and we are working to develop this for other Candida species as well. This was released 2 years ago, and basically what you can do is you can upload your own genomes, and it will rapidly type them in terms of antifungal drug resistance mutations that are known. It will place it in a phylogeny in context with up to 1,000 publicly available Candida auris genomes. And it can also, if you've got the appropriate metadata to say where the location is, it will also plot them on the map. And if there's metadata in addition to that, it can also inform on other things as well. So this is a really nice platform. It's super quick and easy to use, and we really try and encourage people to use this. And we have used genomic surveillance for other outbreaks, the kakapo, for instance. So the kakapo, if I just kind of like talk as I play this, you might have seen this documentary. The kakapo are critically endangered. They are flightless parrots. They're kind of cute. They are quite big, but they're only found on a few islands off the coast of New Zealand. So in 2019, there was an outbreak of avian aspergillosis affecting 21 individuals. And when the population is only a couple of hundred, if there's 9 deaths, then this is really critical. So this was a really interesting project to be involved in. It was a worldwide project. There was us in the UK, obviously the New Zealand team, and then Jason Stike in California. So trying to coordinate meetings was really interesting. And when we performed whole genome sequencing of the strains from the infected kakapo, but also their nests, we could see that the strains differed by only a handful of SNPs, so they were really super related. And it was caused by a single strain that hadn't been previously recorded. So going back to this idea that we are missing diversity, even though that we've sequenced over 1,000 Aspergillus genomes from across the world and over 100 years, we're still missing a certain level of diversity. And we couldn't determine the geographical origin. So we do need more sampling to try and pick up this missing diversity. And then finally, we use genomics to help us keep on top of new emerging fungal species. So as Matt said, this is Trichophyton indotineae, which was originally called Trichophyton metagrophytes variant 8. It causes inflammatory forms of difficult to treat tinea. No one's ever died of this, but obviously it doesn't look very nice, and sufferers do have extreme morbidity. It's terbinafine resistant. The first reports of this were in 2017 in India, and the resistance is due to mutations in the SQLE gene, which is another ERG gene involved in ergosterol biosynthesis. And this is spreading outside of India. It can be transmitted person to person, and there have been reports of it being a sexually transmitted fungal infection as well. So we've looked at sampling across the Northern Hemisphere since sort of 2022. And yeah, so this is where we've come up with. If we compare it to the closely related species Metagrophytes and Indotineae, Indotineae in the red here, this red clade appears incredibly clonal, but also distantly related to Metagrophytes and Indotineae as well. And there doesn't seem to be any clear-cut mutation that's responsible for resistance. So if we look at Indotineae just on its own, there is a lot of diversity within it. It's not as clonal as it originally appears. But the previously reported mutation, this phenylalanine 397 to leucine amino acid mutation, isn't solely responsible for resistance here. So we need to look at the rest of the genome for other markers. Currently, the GWAS, the Genome-Wide Association studies that we've performed on this isn't picking up anything else. So we think that it might not be sort of an amino acid mutation that's responsible. It could be a different mechanism that we are not expecting. I have no idea how long I've been, but to summarize, genomics in mycology has seen tremendous advances. There's been improved short read sequences, but also long read sequencing is becoming more commonplace and accessible and affordable. It can help our understanding of evolution of drug resistance in clinically relevant pathogenic fungi. Genomics helps us identify transmission in epidemiological settings. This has been super useful for Aspergillus fumigatus to help understand the spatio-temporal origins of drug resistance. But it's also super handy for outbreak surveillance of pathogenic fungi, both in Candida auris, but then also integrating this is key for tracking emerging pathogenic fungi. So Trichophyton indotineae is kind of a fungal pandemic, and it would be really crucial to try and integrate this into surveillance systems in public health bodies across the globe. I'll just leave that there, and I'm happy to answer any questions.